Cell death. All cells undergo growth and death. Cellular death is essential for an organism to grow and survive. If cells do not die, or if cells die when they should not, it can lead to or worsen many diseases. Cells die in situations including first, when they are no longer needed, such as during certain stages of development. Second, they die to create a structure in the body. For example, the stratum corneum, which is the outermost epidermal layer, consists of dead cells. Third, they die to control cell number. Excess cells are removed, such as white blood cells, after an infection has been cleared. Lastly, they die as a defensive strategy to remove infected, mutated, or damaged cells. Cells can die because they are damaged, but most cells die by killing themselves. Cell death occurs via different processes. First, apoptosis, second, necrosis, third, necroptosis, and fourth, apoptosis. Cell death proteins. Many proteins have been discovered that control whether a cell dies by the process of apoptosis, necroptosis, pyroptosis, or necrosis. Some key cell death control proteins include caspases, BCL2 family proteins, death receptors, RIP kinases, IAPs, SMAC, or Diablo. Caspases are switched on in apoptotic cells and digest other proteins, which causes cell death. They serve as primary effectors during apoptosis to proteolytically dismantle most cellular structures, including the cytoskeleton, cell junctions, mitochondria, endoplasmic reticulum, Golgi, and the nucleus. BCL2 family proteins interact with each other to determine whether a cell will undergo apoptosis or stay alive. Some BCL2 family proteins promote survival and block apoptosis. Others trigger apoptosis. RIP1 kinase and RIP3 kinase are two proteins that trigger necroptosis. APs, or inhibitor of apoptosis proteins, can prevent cell death by blocking several cell death proteins, including caspases and RIP1 kinase. SMAC, or second mitochondria-derived activator of caspase protein, also known as Diablo, or direct inhibitor of apoptosis binding protein with low PL, is an activator of IAPs. In healthy cells, SMAC is stored in the mitochondria away from IAPs. When cell death is triggered, SMAC can leak out and block IAP's function. Thus, the release of SMAC can promote cell death. Necrosis. Necrosis is the unregulated form of cell death resulting from damage to cell membranes and loss of ion in homeostasis. When damage to membrane is severe, lysosomal enzymes enter the cytoplasm and digest the cell, giving rise to a set of morphological changes described as necrosis. Plasma membrane of the cell constitutes the structural and functional barrier which separates the intracellular from the extracellular environment. Cellular contents leak through the damaged plasma membrane into the cellular space. Then the body recognizes these elements as potential injurious agents and launches acute inflammatory response in the site of necrosis in order to undergo a healing process. Necrosis is always a result of a pathological condition. Morphological appearance of necrosis is the result of two concurrent processes. Enzymatic digestion of the cell. Digestive enzymes are derived either from the lysosome of the dead cells, or autolysis, or from the lysosome of infiltrating leukocytes, heterolysis. Causes of necrosis. Poisons. Poisons that include chemical poisons, toxins of pathogenic organisms, plant poisons, and animal poisons. Chemical poisons may be strong acids and alkalis, drugs, insecticides, fungicides, and other toxic chemicals like lead, arsenate, and phenol. They cause cell death by coagulation of proteins or by poisoning the enzymatic systems. The toxins produced by bacteria, viruses, fungi, rickettsia, protozoa, and metazoan parasites may cause necrosis. Alkaloids from plants also produce necrosis. Cantharidin from beetles and bee stings cause focal necrosis of tissue. Disturbances in circulation. Ischemia or loss of blood supply results in necrosis and infarction. Passive hyperemia causes necrosis when it persists. It is commonly observed in torsion, volvulus, and strangulation of the intestine. General anemia may result in necrosis of the brain and liver if the amount of oxygen and nutrient is not sufficient to maintain cellular metabolism. Mechanical injuries cause necrosis when tissues are crushed or when blood supply is injured or destroyed. Both heat and cold can coagulate protein and cause necrosis. Electric current produced by lightning or generators may coagulate or char tissues. High voltage electric currents cause coagulation of protoplasm. X-ray and nuclear fission substances cause protoplasmic alterations that result in necrosis. The macroscopic appearance of necrosis. The areas are white, gray, or yellow in color. It appears as if the tissues were coagulated or cooked and stand out distinctly from the surrounding normal tissue. Their borders are sharply demarcated and are usually surrounded by a red zone of inflammatory reaction. Abscesses may form if pyogenic bacteria is present. When putrefactive bacteria enter, gangrene may occur which produces additional color changes of green, orange, or black as a result of iron sulfide formation. The microscopic appearance of necrosis. Necrotic cells shows increased eosinophilia. They stain more intensely pink with eosine than normal cells because of the loss of normal basophilia imparted by RNA in the cytoplasm in part to the increased binding of eosine to denatured intracytoplasmic proteins. Cell outlines are indistinct or absent and may have a more glassy homogeneous appearance due mainly to the loss of glycogen particles. The cytoplasm becomes vacuolated and appears moth-eaten when lysosomal enzymes have digested the cytoplasmic organelles. Calcification of the necrotic cells may occur. The nucleus changes in necrosis and characteristics of this change are determined by the manner in which its DNA breaks down. Nuclear changes appear in the form of three patterns. First, the karyolysis. Basophilia of the chromatin may fade, presumably due to the action of DNases of lysosomal origin. Second is the pycnosis. It is characterized by nuclear shrinkage and increased basophilia. The DNA condenses into a solid shrunken mass. Then the karyorexis. The pycnotic nucleus undergoes fragmentation. In a day or two, nucleus in the necrotic cell completely disappears. It also involves the breakup of the chromatin into unstructured granules. The four types of necrosis. Coagulative necrosis. It is the most common type of necrosis and is characterized by preservation of the basic structural outline of the coagulated cells or tissues for a span of at least some days. Cellular detail, however, is lost. This pattern results from denaturation of structural proteins and enzymic proteins due to cell injury or from the subsequent increase in intracellular acidosis, and so blocks the proteolysis of the dead cells, and as a result, enucleate cells may persist for days or weeks. Necrotic cells are removed by phagocytosis of the cellular debris by infiltrating leukocytes and by digestion of the dead cells by the action of lysosomal enzymes of the leukocytes. A localized area of coagulative necrosis is called an infarct. The process of coagulative necrosis with preservation of the general tissue architecture is a characteristic of hypoxic death of cells in all tissues except the brain. Causes of coagulative necrosis: infection, ischemia, mild irritant, heat, or trauma. Macroscopic appearance. Necrotic area is firm and rather dry in consistency. It has a homogeneous, opaque, and cooked appearance, gray, white, or tan in color. And while microscopic appearance shows architectural outline of the tissue or organ when maintained, but cellular detail is lost. Next is the liquefactive necrosis. It denatures the structural protein only. It is a result from the action of powerful lysosomal enzymes, and these enzymes digest the dead cells and transform the tissue into liquid or viscous mass. It is a characteristic of focal bacterial infections because these agents constitute powerful stimuli to the accumulation of neutrophils. Enzymes of bacterial and leukocytic origin contribute to the digestion of dead cells. This is the type of necrosis observed in abscesses and suppurative wound infections. Necrotic material is frequently creamy yellow because of the presence of dead leukocytes and is called pus. Hypoxic death of cells within the central nervous system often manifests as liquefactive necrosis. Cause of liquefactive necrosis is pyogenic organisms. Macroscopic appearance. Tissue in the area of necrosis is liquefied and may be watery, tenacious or semi-solid in consistency. The color is usually white, yellow, or red. The connective tissue wall may be formed around the necrotic mass if the necrosis is of long standing. Microscopic appearance. There is no architectural outline or the cellular detail is visible in the area of necrosis. The dead tissue is homogeneous and stains pink with eosin. The entire necrotic mass is surrounded by a zone of acute or chronic inflammation depending on the length of time necrosis has been present. Next, caseous necrosis. It is characterized by the conversion of dead tissue into a homogeneous granular mass resembling a cheese, and by the absence of both architectural and cellular detail. It is caused by chronic infections like Mycobacterium tuberculosis and systemic fungal infections. Macroscopic appearance. The mass is dry but creamy in consistency. It is soft, friable, and white-gray in color. The calcium salts are frequently deposited in the dead tissue. Caseous mass is enclosed within a connective tissue capsule. Microscopic appearance. Necrotic focus is composed of structureless, amorphous granular debris enclosed within a distinctive ring of granulomatous inflammation. There is no architectural or cellular detail present. Calcification commonly occurs in the necrotic areas, especially in sheep and cattle. A fat necrosis. It is the death of adipose tissue within the living individual. For fat necrosis, there are three types. First, the pancreatic fat necrosis. Pancreatic fat necrosis is the death of adipose tissue in the vicinity of the pancreas due to the action of lipases. It is caused by some injury to the pancreas or its duct. Primary or secondary tumors of the pancreas may also induce this necrosis. Powerful lipases released from the pancreas destroy not only the pancreatic substance itself, but also the adipose tissue in and around the pancreas and throughout the peritoneal cavity. The released fatty acids combined with calcium produce grossly visible chalky white areas. It appears as white or yellowish white, chalky, opaque masses in the interstitial adipose tissue of the pancreas and the peripancreatic fat. A zone of acute or chronic inflammation appears around the necrotic areas. Connective tissue may undergo metaplasia and produce a bone. In a microscopic appearance, only shadowy outlines of necrotic fat cells may be seen, with basophilic calcium deposits and a surrounding inflammatory reaction. Next type of fat necrosis is the traumatic fat necrosis. It is the death of adipose tissue in an area of mechanical injury. It commonly occurs in the subcutaneous adipose tissue due to mechanical injuries during working, fighting, or exercising. Third type of fat necrosis is the nutritional fat necrosis. Necrobiotic alteration in fat that is associated with extreme emaciation and debility. It occurs in starving or debilitated animals. It may occur throughout the body but is most common in the abdominal fat. Chronic inflammatory reaction occurs at the junction of the necrotic and living tissue. Macroscopic appearance. Fat is opaque, chalky white, and usually firm. It may undergo calcification. Microscopic appearance. Adipose cells contain a pale pink, slightly granular material in which numerous clefts and crystals are seen. When calcification occurs, the small spherical masses present stain blue with hematoxylin. Let's move on to gangrene. Gangrene is the invasion and putrefaction of necrotic tissue by saprophytic bacteria. Putrefaction means decaying or rotting. Saprophytic bacteria are bacteria that breaks down or decompose organic matter like E. coli and spirochetes. Gangrene is most often seen in the lungs, intestine, and extremities. In the lungs, it occurs most commonly from faulty drenching of medicines. Then, in the intestines, it is a result of a verminous thrombus in the anterior mesenteric artery. And in the extremities, it is associated with freezing. Types of gangrene. First is the dry gangrene. Dry gangrene occurs at extremities like tail, tip of ears, tip of scrotum. Due to necrosis and invasion of saprophytes, the evaporation of moisture takes place resulting into dry lesions. Dry gangrene happens when circulation is no longer maintained and when the coagulative necrosis is dominant. Next type is the moist gangrene. Moist gangrene mostly occurs in the internal organs of the body like lungs, intestine, and stomach. Moist gangrenous tissues appears to be red, green, gray, black in color as a result of sulfide formation. The last type of gangrene is the gas gangrene. Gas gangrene occurs in muscles, particularly of thigh muscles of the hind legs, in heifers in case of back leg. Gas gangrene is a specific type of moist gangrene due to invasion of clostridial organisms. Clostridial organisms are inhabitants of the soil and the digestive tract. They enter into the wounds by shearing, castration, docking, and ear notching.
Post-mortem change. Simply defined as alterations in cells or tissues occur after death of animal. The degree and pace of such changes are determined by the factors of one set. The first factor is the temperature in the environment. Since increased summer temperatures increase the rate of enzymatic and bacterial activity. Animals decompose very rapidly in the summer. Animals can be preserved for long periods with modern refrigeration. The next factor is the animal's size. The larger the animal, the more rapidly will post-mortem change occur. This is because the large animal require a longer period of time before the body heat is dissipated. The next one is the external insulation. Thick cutaneous coverings of fur, feathers, hair, or wool prevent heat dissipation. As such, sheep decompose very rapidly because of insulating wool. Apart from the size and insulation, the species of animals also determines the character of the flesh. For example, pig flesh is soft, moist, and contains fat. Therefore, the rate of decomposition is rapid. On the other hand, horse flesh is dry and rather firm. Therefore, the decomposition is relatively slow. And lastly, nutritional status. Fat is an insulating substance. The fatter the animal, the slower will be the loss of heat and the more rapid will be the rate of decomposition. So let us proceed to the postmortem change, which includes autolysis. Autolysis refers to the digestion of tissues by their own cellular enzymes. When tissue is fixed or embalmed, cellular enzymes are inactivated by formalin or alcohol and autolysis is prevented. The next is the putrefaction. It is the decomposition of tissues by enzymes of saprophytic bacteria. So, after autolysis, the saprophytes invade from external environment into the body, and then it multiply and eventually digest the tissues with their enzymes. Therefore, the tissue becomes fragile and it produces foul odor. The next one is the rigor mortis. It is the shortening and contraction of muscles that occur after death and result in stiffness and immobilization of the body. The rigor mortis begins in the anterior portion and progresses in a posterior direction, such as the head, the neck, the trunk, and the limbs. Usually, it appears after 1 to 8 hours after death. In emaciated animals, it appears slowly. It is never pronounced and may not appear at all. The next one is the livor mortis. It is the staining of tissues with hemoglobin after death of animals. It gives pinkish discoloration to the tissues. Moving on to the next, the algor mortis. Algor mortis is the cooling of the body because there is no circulation of blood after death, which keeps the body temperature stable. The body cools down after death. However, depending on the species, environmental temperature, and type of animal, it can take 2 to 4 hours. Moving on to the next post-mortem change, the post-mortem clotting of blood. It is the coagulation of blood in vessels after death. In dead animals, the endothelial cells begin to degenerate and liberate thromboplastin, which then clots the blood within the heart, arteries, and veins. In anthrax, however, no clotting occurs because fibrinolysin produced by the bacteria liquefies fibrin. In sweet clover poisoning, also clotting does not occur since prothrombin activity is inhibited. Moving on to the next postmortem change, the imbibition with hemoglobin. It is the staining of tissue with hemoglobin. After death, the erythrocytes are hemolyzed by cellular and bacterial enzymes, and hemoglobin is liberated. Being soluble in body fluid, it diffuses into the surrounding tissues and stains them red. The next post-mortem change is the hypostatic congestion. It is simply defined as the accumulation of blood in the ventral portion of organs and the entire carcass due to the influence of gravity. Next is the pseudomelanosis. It is the appearance of gray, green, or black pigment in the tissues after death. Hydrogen sulfide produced during putrefaction combines with the iron to form iron sulfide, a black pigment. The concentration of this pigment and its combination with other tissue pigments result in a variety of shades of green, gray, and black. After the pseudomelanosis, the imbibition with bile is the next postmortem change. Imbibition with bile is the yellow pigmentation of tissue occurring near the gallbladder when bile pigments diffuse into the surrounding tissue. The next is the postmortem emphysema. It is the accumulation of gas in tissues as the result of bacterial fermentation. It occurs due to decomposition by gas-producing organisms including saprophytes. The gas is mainly accumulated in gastrointestinal tract, causing rupture of the organ. The rupture of organs and tissues occurs when gases produced cause progressive distension of body structures until they burst. Occurs in stomach, intestine, diaphragm, and ventral abdominal wall. The rupture of organs and tissues occurs when gases produced cause progressive distension of body structures until they burst. Occurs in stomach, intestine, diaphragm, and ventral abdominal wall. The last postmortem change is the displacement of organs. It occurs when the dead animal is rolled over or moved. The intestine is usually displaced. Pathological calcification. It is defined as the deposition of calcium phosphates and calcium carbonates in soft tissues other than bones and teeth. Its concentration in the blood fluctuates within rather narrow limits and in general averages about 10 mg per 100 ml of blood. Pathological calcification can be classified as dystrophic and metastatic calcification. Dystrophic or local calcification, defined as the deposition occurs in dead or necrotic or dying tissues. It occurs in a focal area of the body. Dystrophic calcification is associated with many diseases and occurs in areas of necrosis, whether coagulative, caseous, or liquefactive type. Also in foci of enzymatic necrosis of fat. One of the most common causes is tuberculosis. Calcium is deposited in the caseous tissue of the tuberculous lesion. Macroscopically, the calcium salts appear as fine, white granules or as yellowish-white or gray chalky masses within the tissue, often felt as gritty deposits. Calcified tissue has a firm consistency, and when cut with a knife, a definite grit can be detected against the knife blade. Calcification is especially common in cattle, sheep, and rabbit. Microscopically, with the usual H&E stain, calcium salts appear as deep blue granules or masses. They can be intracellular, extracellular, or in both locations. A more specific staining technique is von Kosa's silver nitrate stain that stains the calcium salts as black spears or masses within the tissue. The next type of pathological calcification is the metastatic or general calcification. It is characterized by deposition of calcium salts in soft tissue, and it almost always indicates some derangement of calcium as a result of hypercalcemia. Hyperparathyroidism is associated with a parathyroid neoplasm. The increased parathyroid activity removes calcium from the bones that become rarefied and large cyst-like spaces appear with them. Likewise, myeloid tumors in the bone marrow and metastatic cancer cause demineralization of the skeleton, and the bloodstream is flooded with calcium. Also, vitamin D-related disorders, including vitamin D intoxication. Hypercalcemia may also arise in renal diseases, particularly in advanced renal failure. The deposition of calcium salts may occur in organs throughout the body, especially in those that secrete or excrete acid substances such as the stomach, the kidney, and the lungs. The macroscopic and microscopic features of metastatic or general calcification are organ becomes hard. Wall of arteries becomes hard due to calcium deposits. Deposition of calcium in soft organs like myocardium, arteries, muscles, and etc. Purplish or black color calcium surrounded by comparatively normal tissue. Morphologically, the calcium salts in all these sites resemble those described in dystrophic calcification.
Apoptosis. It is a Greek word meaning a falling away from. Apo means away and ptosis means falling. Apoptosis is a form of cell death designed to eliminate unwanted host cells through activation of a coordinated, internally programmed series of events brought about by a set of gene products. Meaning, it is a physiological response so that the body can prevent accumulation of diseases. It can also be described as the programmed cell death in several important physiological and pathological processes. It is a distinctive form of cell death manifested by characteristic chromatin condensation and DNA fragmentation. It means the cell will undergo shrinkage or its function is the deletion of cells in the normal development, organogenesis, immune function, and tissue growth, but it can also be induced by pathological stimuli. The following are the physiological and pathological processes that involve apoptosis. First is the programmed destruction of cells during embryogenesis as occurs in organogenesis. Embryogenesis is the development of embryo, while organogenesis is the development of organs. Second is the hormone-dependent physiological involution, such as involution of endometrium during menstrual cycle in humans, or the lactating breast after weaning. Endometrium is which houses the baby during pregnancy. And second example is the pathological atrophy, or the prostate after castration in humans. Dun sa lactating breasts na kailangan mag-produce ng milk, magpapakamatay or papatay na yung mga milk-making cells or alveoli, thus apoptosis occurs. Third is the cell deletion in proliferating populations such as intestinal crypt epithelium or cell death in tumors. Kailangan nitong mangyari para maiwasan yung pag-accumulate ng tumor na pwedeng mag-cause ng cancer. Fourth is the death of neutrophils during an acute inflammatory response. Neutrophil fights infection by ingesting microorganisms and then killing them. Fifth is the deletion of autoreactive B and T cells. T cells are involved in cell-mediated immunity, whereas B cells are primarily responsible for humoral immunity. This happens using a protein called perforin. Sixth is a variety of injurious stimuli that produce necrosis, but when given in low doses, produces apoptosis. An example of this is the use of cytotoxic anti-cancer drugs, wherein it purposely induces cell death to kill infected cells. But when it is given in high doses, necrosis will occur or take place. Kasi kung dahan-dahan lang yung pagpatay ng mga cells, mas mafo-focus ito dun sa mga infected cells. Pero kung mabilis yung pagpatay or high dosage ang gagamitin, makaka-harm na siya sa organism. Thus, failure of cells to undergo physiological apoptosis may result in abnormal development, unhindered tumor proliferation, or autoimmune disease. Ibig sabihin, apoptosis can also be a form of immunity. The following are the morphological features of apoptosis. First is the cell shrinkage. The cell is smaller in size, the cytoplasm is dense, and the organelles are more tightly packed. Ibig sabihin, yung cell is liliit, tapos yung mga organelles is magkakalapit-lapit sila sa isa't isa, and yung cytoplasm, liliit din. Next is the chromatin condensation. This is the most characteristic feature of apoptosis. Ito yung part na yung DNA is magfo-form siya ng mas maliit and mas makapal na coil para mas mag-fit siya dun sa cell kasi nga 'di ba liliit yung cell. The nucleus itself may also break up producing two or more fragments. Ibig sabihin, yung original cell magiging mas maliliit na siya or maghihiwa-hiwalay. Next is the formation of cytoplasmic blebs or buds or yung curves na makikita sa surface ng cell habang lumiliit siya. And then, apoptotic bodies or yung result ng paghiwa-hiwalay ng cells into smaller pieces. Phagocytosis of apoptotic cells or bodies by nearby healthy cells, either parenchymal cells or macrophages. This is the part wherein phagocytes will ingest apoptotic bodies so that the result of apoptosis, which is the smaller pieces of the original cell, will be cleaned up. The following are the four mechanisms of apoptosis. First is the signaling, next is the control and integration, next is the execution or death, and finally is the removal of dead cells. For the signaling, apoptosis may be triggered or initiated by a variety of stimuli. Here are the following stimuli which can be observed. Intrinsic embryo signals, selected injurious stimuli, a lack of growth factor, specific receptor-ligand interaction, and release of granzymes from cytotoxic T cells. If any of these are recognized, the cell will undergo apoptosis. For the control and integration, this is performed by specific proteins. Proteins connect the original death signals with the execution program. They are important because their actions may result in either success or failure of original death signals. Apoptosis is controlled by proteins depending on the situation. For example, for cell-mediated response, it deletes infected cells using a protein called perforin. There are two broad pathways of control and integration. First is direct transmission of death signals by specific adapter proteins to execution mechanism. Ibig sabihin, pagka-recognize ng protein, diretso agad siya sa execution. An example of this is phospholigand model and target cell killing by cytotoxic T lymphocytes. Next pathway is it involves members of the BCL2 family of proteins. BCL2 family is situated in the outer mitochondrial membrane. BCL2 binds with pro-apoptotic proteins, thus inhibiting apoptosis. There are two ways wherein BCL2 suppresses apoptosis. First way is by direct action on mitochondria. It prevents increased mitochondrial permeability. Second is by effects mediated through actions with other proteins. So, ano nga ba yung mitochondrial permeability? Formation of these pores in the inner mitochondrial membrane reduces membrane potential, resulting in diminished ATP production and mitochondrial swelling. So, kapag yung mitochondria, hindi na siya nakakapag-produce ng ATP, which is the energy to power the cell, hindi na siya mag-work ng maayos. Kaya, wala nang silbi yung cell, thus, kailangan na niyang mag-undergo ng apoptosis. The signals also cause increased permeability of outer mitochondrial membranes, releasing the apoptotic trigger, cytochrome C, from mitochondria into cytosol. Ibig sabihin, mas madali nang makapasok yung mga ibang substances sa loob ng mitochondria, katulad ng cytochrome C, which is an initiator of apoptosis. For the third mechanism, which is execution or death, this is the final pathway of apoptosis, which is characterized by certain distinctive biochemical changes. Some of these events may be seen in necrosis, but other alterations are more specific of apoptosis. These events result from the synthesis and activation of a number of cytosolic catabolic enzymes. There are three final execution pathways. The first final execution pathway is protein cleavage breakup. A specific feature of apoptosis is protein hydrolysis. This is brought about by a newly discovered family of cysteine proteases called caspases. Caspase C, wherein the C indicates a cysteine protease mechanism. That is, they all have the amino acid cysteine in their active site, while aspase refers to their unique ability to cleave or split after aspartic acid residues, which means that the protein will cause the breakup of the cell. Next is the protein cross-linking. Extensive protein cross-linking brought about by transglutaminase activation converts soluble cytoplasmic proteins into a covalently linked condensed Rankine cell, which then readily breaks into apoptotic bodies. Kung yung kanina yung protein is maghihiwa-hiwalay, dito yung protein is magbabind together hanggang sa maghiwa-hiwalay sila dahil sa sobrang pag-condense. The last execution pathway is DNA breakdown. Apoptotic cells exhibit a characteristic breakdown of DNA into 180 to 200 base fragments. This is brought about by the action of calcium and magnesium dependent endonucleases. This simply means that the breakdown of DNA into smaller pieces will result into the death of a cell. The final mechanism of apoptosis is the removal of dead cells. Apoptotic cells express phosphatidylserine in the outer layers of their plasma membrane. Phosphatidylserine can be observed in the surface of apoptotic bodies. It is like a signal for phagocytes so that they can undergo phagocytosis. In some types of apoptosis, thrombospondin, an adhesive glycoprotein, is also expressed in the surface of apoptotic bodies. Just like phosphatidylserine, thrombospondin is a signal so that they can undergo phagocytosis. These changes permit the early recognition of dead cells by macrophages and nearby cells for phagocytosis without the release of mediators or inflammation. Ibig sabihin, sa phagocytosis, unlike sa necrosis, hindi na kailangang mamaga ng mga cells or lumaki. Kailangan na lang hanapin ng mga phagocytes kung aling dead cells ang may phosphatidylserine or thrombospondin para malinis sila kaagad. In this way, the apoptotic response disposes of cells with minimal damage to the surrounding tissue. The process is so efficient that dead cells disappear without leaving a trace and inflammation is virtually absent. The following are specific examples of apoptosis. First is signaling by tumor necrosis factor or TNF family of receptors. Second is cytotoxic T lymphocyte stimulated apoptosis. Third is apoptosis after growth factor withdrawal. Fourth is DNA damage mediated apoptosis. Signaling by tumor necrosis factor or TNF family of receptors. Tumor necrosis factor is a cytokine or a small protein used by immune system for cell signaling. As I've mentioned earlier that apoptosis is controlled or integrated by proteins. This is an example of that. The TNF-R family includes members that bind not only the important cytokine TNF but other significant ligands as well. Some initiate apoptosis, some cell proliferation, and others initiate both. A specific example of this is Fas ligand mediated apoptosis and TNF-induced apoptosis. For the Fas ligand mediated apoptosis, this type of apoptosis is called by a cell surface receptor called Fas or CD95. A membrane-bound ligand called Fas ligand, produced by cells of the immune system, binds to Fas on T cells and activates a death program. So, if the Fas ligand and T cells bind together, apoptosis will start and act as an immune response, thus preventing diseases. This system is important in eliminating lymphocytes from an immune reaction, thereby limiting the host response. As for the TNF-induced apoptosis,
Activation of one of the TNF receptors by cytokine TNF can lead to apoptosis by causing association of receptor with the adapter protein TRAD. TNF is secreted by inflammatory cells or infected cells which may be involved in inflammation-associated carcinogenesis. So if a cell secretes TNF, it's a signal that it must undergo apoptosis. For the cytotoxic T lymphocyte-stimulated apoptosis, this is the type of apoptosis which is caused by cytotoxic T lymphocyte or CTLs. CTLs recognize foreign antigens on the surface of host cells. Ibig sabihin, kapag na-recognize nila na yung cell is hindi normal yung behavior or may nakapasok na foreign matter, kailangan mag-undergo ng self-destruct ng cell para hindi siya maka-affect pa ng sobra. Apoptosis after growth factor withdrawal. In the absence of growth factor, the cells undergo apoptosis. On withdrawal of growth factor, pro-apoptotic members of the BCL2 family proteins move from the cytosol to the outer mitochondrial membrane and change the ratio of pro-apoptotic and anti-apoptotic members. So, ibig sabihin, kapag hindi na nagre-release ng growth factor yung cell, mas dadami na yung pro-apoptotic members. DNA damage-mediated apoptosis. Exposure of cells to radiation or chemotherapeutic agents induces apoptosis by damaging DNA and activating tumor suppressor gene TP53. In chemotherapy, radiation is given at a mild dosage and controlled manner. But if the exposure to radiation is too much, that falls under necrosis. TP53 accumulates when DNA is damaged and arrests the cell cycle to allow additional time for repair. However, if the repair process fails, T53 triggers apoptosis.
Necroptosis. Necroptosis is an alternative mode of regulated cell death mimicking features of apoptosis and necrosis. Necroptosis has the same goal as apoptosis and necrosis, which is to kill the cell, but its mechanisms are different. It is triggered by outside trauma or deprivation compared to apoptosis, which can start from signals within the cell. Necroptosis results in innate immunity by actively including cell death of infected cells or by provoking the immune system via the release of danger signals to clear pathogens. Necroptosis is activated by the mediation immune ligands including FAS, TNF, and LPS, leading to activation of RIPK3, which further activates the MLKL by phosphorylation. Phosphorylated MLKL translocates into the inner leaflet of the plasma membrane and disturbs the integrity of the cell, in which MLKL is the protein required to damage the membrane in necroptosis. Pyroptosis. Pyroptosis is another form of cell death that is triggered by pro-inflammatory signals and associated with inflammation. This type of cell death is seen primarily in inflammatory cells such as macrophages and may be triggered by bacterial or pathogen infections. In pyroptosis, it is observed that bacteria-infected macrophages underwent a rapid lytic cell death dependent on caspase-1 activity. GSDMD is the protein required to damage the membrane in pyroptosis.
Cells that undergo pyroptosis display morphological characteristics such as cell swelling, membrane blebbing, DNA fragmentation, and eventual cell lysis. The nucleus, however, remains intact.